Welcome to Life and Biology. I'm Dr. Joel Graff. This video is a condensed lecture for my Discover Biology class. That's a Biology 101 class at Montana Tech. We have Non-Mendelian Genetics is the topic today, and if we're going to talk about non-Mendelian genetics, it's probably good to do a quick review of Mendelian genetics. So in Mendelian genetics, here we go, we have a crossing of two organisms to see what their offspring look like. In this case, we have one parent which has the genotype A, and we have another parent which has a genotype of lowercase a. Now the capital letter means that you're talking about a dominant trait. The lowercase letter means you're talking about a recessive trait, and this is of course based on Mendel's project. When he worked with the pea plants, he looked at 7 different traits of the peas, and one of them was flower color, so we're just going to go ahead with flower color as our example here. The dominant trait for flower color is purple. The recessive trait for flower color is white. So a capital A would be a purple flower. Lowercase a would be a white flower. And if you take across those two organisms, this first parent can make a gamete with a capital A or another gamete with a capital A. It's homozygous, so it only gives you one option, but I went ahead and wrote them all out. Then we've got lowercase a for the other parent, so I put that here and here. Then I mix and match one gamete from this parent and one gamete from that parent, and I get a capital A, lowercase a for that square, and that square, and that square, and that square. And so those are heterozygotes. They're heterozygous for the A gene. And so for a genotype in this cross between two what we call true breeding parents is 100% heterozygous, capital A, lowercase a. And the phenotype for that, because we're dealing with a dominant and recessive trait, it's 100% of the dominant trait is going to be seen in all of these heterozygotes, and that again was the purple color. So let's take this set of offspring and cross them with each other. So we've got a heterozygote crossed with a heterozygote, and this is a monohybrid cross. I think on the previous video I might have called it a dihybrid cross, so that's kind of embarrassing. It's mono. We're dealing with one trait, one type of hybrid, the A and the capital A, lowercase a. So this first parent, it can make gametes with a capital A, or it can make gametes with a lowercase a. The second parent, same thing. It can make gametes with a capital A or gametes with a lowercase a for this particular gene. And we go ahead and match up this gamete, capital A, with this gamete, capital A, and the offspring would be homozygous for capital A. We've got a heterozygote here because we got a capital A gamete gene from that parent and a lowercase a gamete for that. Heterozygous again for the same reason. We have a capital A and a lowercase a that were given by the parents to make this type of kid. And then we've got lowercase a. This 1 out of 4 children should get that combination. There's a 50-50 chance of getting this lowercase a and a 50-50 chance of getting this lowercase a, and so the chance of getting those two at the same time is 50% times 50%, and so that's 25%. That's why it takes up 1 out of 4 squares. So anyway, genotype. 25% are homozygous for the dominant, 50% are heterozygous, and 25% are homozygous for the recessive. For our phenotype, whether you have 2 A's or 2 capital A's or 1 capital A and 1 lowercase a, you're going to display the dominant trait, which was purple. So 75% of the monohybrid cross would show the dominant trait, and 25% of the monohybrid cross would end up with the lowercase a, and that means that 25% of the offspring would show the recessive trait. So this 1 out of 4 would be white, and these 3 out of 4 here are going to be purple. So that's what a dominant trait looks like, and that's Mendelian genetics. But again, the focus of this lecture is about dominant or non-Mendelian genetics. So now that we've gone over Mendelian genetics, let's do some compare and contrast with what non-Mendelian genetics would look like. And this is not a complete listing of all the different non-Mendelian genetic things we could talk about, but it's a good start for 100 level biology class. So under Mendelian genetics, we've got dominant and recessive, where we had the capital A and the lowercase a, and we had pointed out that if you're homozygote, if you're homozygous for the, let's say, flower color, purple or white, you would show the dominant trait, which was purple. In non-Mendelian genetics, I have a 1A and a 1B to match up with this one, and that's because there's a couple different things that can happen with dominance. One, you can have incomplete dominance, and the other is to have codominance. I'll go through each of these on a slide of their own. So moving on to point number 2 for Mendelian genetics, you have 2 alleles per trait. That is, you could have a purple allele or a white allele. Those are your only options that you can deal with. We only dealt, on the previous example for Mendelian genetics, we only dealt with a capital A for the purple allele and a lowercase a for the white allele. We didn't throw in any other alleles like say a Greek alpha or something like that. So you only deal with 2. And another example from Mendelian genetics was round and wrinkled for the peas in the pea pod. Okay, another one, so the counter to having 2 alleles per trait, obviously, is many alleles per trait. So individuals only have 2 alleles because they get 1 allele from their mother and 1 allele from their father. But if we were to look in the whole population of, say, people or plants, whatever it is our test subject is, the entire population would have more variety than just 2 possible alleles. Number 3, independent assortment. This is when each trait was independent of the other traits as far as how it got sorted into the gametes. And I didn't show that in the previous video. The classic example of Mendelian traits and showing an independent assortment is a dihybrid cross, and so I will show that in this video. Conversely, in the last video we talked about genetic linkage, and that would be example of non-Mendelian. That is when the 2 traits don't or tend to travel together when the gametes are made, and so that usually comes from the genes being close together on a chromosome. Okay, number 4, traits are on somatic chromosomes. Okay, so in humans we have 46 chromosomes total per cell. It's 23 pairs of chromosomes. Those pairs are called homologous chromosomes. 22 out of the 23 are somatic chromosomes, and which leaves you with just what's that 23rd set of chromosomes, and those are sex chromosomes. And so you've probably heard of X and Y chromosomes. If you have 2 X's, you'll have a female. If you have a X and a Y, you'll have a male. And then the thing about the X chromosome is that it's a lot larger than the Y chromosome. When we matched up these pairs of somatic chromosomes, they're basically the same length each, but the X is way longer in number of nucleotides than the Y, and so more traits, more genes are going to be found on the X chromosome than the Y. And so when you talk about sex-linked traits, most of those are going to be X-linked traits. The fifth point about Mendelian genetics to bring up at this point is that there's one gene per trait. So when we were determining what color the flowers were going to be on the peas, there was just the gene that was the capital A or the lowercase a. So there, that one gene, A, we'll call it, controlled the flower color. In non-Mendelian genetics, you have the possibility that more than one gene contributes to a given trait, and we'll go through the classic example of height when we get to that one. Okay, jumping into incomplete dominance. When we talked about Mendel, we talked about pea plants and that the purple gene, capital A, was dominant to the white gene, which was the lowercase a. So if you have a heterozygote, capital A, lowercase a, the first one is going to be the capital is going to be the trait that you see. So a heterozygote would be purple. Snapdragon flowers are different. The flowers can be red or white or pink. That should be an or. And so we have an allele for red, which is a capital C with a superscript R, and we have an allele for white, which is capital C superscript W. And we'll run through, let's say you have true breeding red snapdragons and you cross them with true breeding white snapdragons. We do our Punnett square like we did with the Mendelian genetics, and we mix and match one gamete from this parent, one gamete from this parent, and then we fill out our 4 squares of our Punnett square. And in this case, 100% are heterozygous, and that's what we had after a cross with true breeding things in the Mendelian genetics example a couple slides ago. But this time, rather than 100% of the plants being red, we have 100% of the plants being pink, so midway between red and white. Okay, so those heterozygotes that you got from this cross, these offspring, if we were to cross those with each other, which is what I have here, C to the uppercase R and C to the uppercase W, crossed with C to the uppercase R, C to the uppercase W. So that, remember, this would be the F0 or parental generation. These offspring, either if you want to look there or there, are the F1. And then if we cross those, these are the 2 possible gametes, or 2 possible alleles you could get in your gametes for this trait from this parent, and these are the 2 possible genes you could get in the gametes from this parent. And in this case, we have our first square is C to the uppercase R, C to the uppercase R, and we know that that was red. Here was an example of red before. In this square, we've got a heterozygous C to the uppercase R, C to the uppercase W. Same thing in this square. And in those cases, we just learned that if you are a heterozygous, you'd be pink. And then the C to the uppercase W, C to the uppercase W is when you would have a white plant. So in the case of snapdragons, your F2 generation would look like this, assuming you started with a cross between true breeding red and white snapdragons. So because red and white, because neither one is the completely dominant over the other, this is considered incomplete dominance, and you get kind of a merger of those 2. Okay. Next, codominance and also multiple alleles. So just jumping back to, we have codominance was 1B and multiple alleles was number 2. So when we talk about ABO blood groups, we're going to kill 2 birds with 1 stone, basically, because we're going to talk about codominance and we're also going to talk about multiple alleles. So what are those multiple alleles that we can work with? For ABO blood groups, if you have a capital I, to the uppercase A and a capital I to the uppercase B, and then you can have a lowercase I. This first one to the uppercase A, the cells would have the A protein on their surface, and this is in blood cells. The B would have I to the B would make the cells express a protein called B on their surface. And then if a cell had the lowercase I, it wouldn't make any protein from that. And so in this case, sticking with our capital letter and lowercase letter like Mendelian genetics, each of these alleles with the capital I is dominant to the lowercase I. But something happens when you have the codominance occurs when you have the capital I matched up with another capital I. So let's look at the genotypes that are possible here. Capital I to the A, capital I to the A, so homozygous for that first allele would give type A blood. Capital I to the uppercase A, and then a lowercase I, which won't make any extra protein. Those cells still will have the A protein being expressed on their surface, so the person with is heterozygous this way is still type A blood. The capital I to the B, similar to capital I to the A, except for a homozygous would be type B blood. And then here's our heterozygous, where the second allele is the recessive allele, or it doesn't express anything, and so those cells would make the B protein on their surface and nothing else, so that'd be type B. So here's codominance comes into play, is when you have capital I to the capital A and capital I to the capital B. People with that particular genotype are going to have type AB blood, because the capital I to the A allele would make those cells express the A protein, and the capital I to the B allele would make those cells express the B protein, and there's plenty of room on a cell surface for both A and B to be expressed, and so those people would have type AB blood. And then finally, if someone is homozygous for the recessive lowercase I, then they wouldn't make either A or B proteins, and so that person is considered type O, neither A or B. Almost think it should be called type 0 since you don't have A or B, but I didn't come up with this. So let's take a look at an example cross. I won't go into all the possible examples that we can do, we'll just go through one. Let's say we have one parent that has type AB blood, so that means that they have capital I to the capital A and capital I to the capital B are the possible genes that the gametes could get packaged with. And the other parent has type A blood because they have capital I to the A, and then they have the lowercase I that doesn't do anything really. So let's look at what kind of offspring you could expect and the ratio you'd expect those offspring to have for blood types. This first square is capital I to the capital A, capital I to the capital A, and that's the only square that has that, so 25% of the offspring from these two parents should have that genotype, and that would be type A blood. Then you have capital I to the A and capital I to the B mixed together, and again that's the only square that gets it, so 25% have that genotype, and that's type AB blood. Then we have 25%, 1 in 4 has a capital I to the A and a lowercase I, and that's the genotype there. I don't know why the I has a weird dot on it, but that is phenotype is type A blood. So we have two different possibilities for type A blood. So if these two individuals had children and you notice that one of their kids has type A blood, you'd still need to do some genotyping to figure out what their exact genotype is, because this A and this A phenotype doesn't let you exactly know what the genotype would be. And the final square in the bottom right corner here is capital I to the capital B. It's terrible handwriting. Lowercase I, and that is blood type would be B. Those cells would express the B protein from this first allele, and it wouldn't express any protein from the second allele, and so it would be the phenotype is type B blood. Okay, so here's where I got things screwed around, or switched around. I had talked about genetic linkage last time, and then this time I'm going to talk about Mendelian genetics. So I want to compare independent assortment versus genetic linkage. So this is tracking. For this sort of idea, you need to track 2 or more traits. Okay, so all of the other ones we've only dealt with one trait at a time. But let's say we have a trait encoded by the A gene and a trait encoded by the B gene, and we have 2 alleles for A. We have a capital A and a lowercase a, and we have 2 alleles for B. That's capital B and lowercase b for this individual. So the A is heterozygous or a hybrid, the B is a heterozygous or a hybrid, and so this is where I should have said dihybrid cross last time. You cross that with a second parent that's a dihybrid, and you cross those two, that's a dihybrid cross. Okay, just to step back, if you've ever done FOIL in algebra where you had a couple numbers in parentheses that you multiplied by a couple other numbers that were in parentheses, you might have learned the FOIL method. And so that was, you take the first times the first, the outer times the outer, the inner times the inner, and the last times the last. So that's kind of the same way here. So let's look at this parent here, and we're going to consider what all combinations of A and B genes you could get from a dihybrid parent like this. So let's take the first and the first, the capital A and the capital B, and I used blue to code for those. Switching over to green would take the outer times outer, so the capital A and the lowercase b. Then we do the inners, so that's the lowercase a and the capital B with the red arrows. And then we have the last and the last, so that's the lowercase a and the lowercase b. So the genotype is shown there. The other parent, I didn't draw it all out, but the other parent would have those same exact possibilities since they're exact same genotype as this first parent. So when we go to make our Punnett square, we have 4 possible gametes that we want to keep track of. And so then we have 4 gametes listed for the first parent here, and 4 gametes listed for the second parent here. And so it's a 4x4 grid where all of our others have been in a 2x2 grid. So we've got 16 squares total. So I did color coding so you can see what I did here, but this first square is easy to explain because it's this gamete and that gamete, and so this blue letters with those blue letters, and then this is the green letters with the green letters, the red letters with the red letters are here, and the black letters with the black letters are here. Then I would just go down, so everything in this column would have the capital A in blue and a capital B in blue. Everything in this row here would have a capital A in green and a lowercase a, or a lowercase b in green, all the way across. And then I just plotted through that sort of thing until I filled out all 16 possible offspring that could come from just these 2 traits. So just like before, we would talk about genotype and phenotype. Before we only had 4 boxes to deal with. Here we've got 16. And when you look at the genotype, that's when you really get, it really sounds confusing. Your ratio is 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. And why do I have it in that order? It's because here's your 1, here's your 2, here's your 1, here's your 2, there and there. Then there's 4 down the middle. 4 of them have the heterozygous at both for both genes, and then you just reverse back out of there. This square and this square are the same genotype. This is a unique genotype. I didn't finish it off there. Let's see. We need a capital B, don't we? Let's add that in. Okay, and then filled them all out. There's 2 that have this particular set of genes, and then there's only 1 that is homozygous recessive for both. And so when we talk about phenotype 9 to 3 to 3 to 1, this one that stands out is this one right here, because it's recessive for both, and that's the only time you could see the recessive trait for gene A and the recessive trait for gene B is that one. Then these 3 here, this one, this one, this one, are going to have the recessive trait for A and the dominant trait for B. So that's where one of those 3 comes from. And then this square, and this square have the dominant trait for A and the recessive trait for B. So again, that was this one, and this one. So that's where one of those 3's come from. And then the 9, it comes from the fact that 9 of these squares are going to have the dominant trait for A and the dominant trait for B. So if you went through and searched, you'd be able to find those. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so that's the old dihybrid cross. Now what would happen if you were studying some genes that were genetically linked? Your ratio of phenotypes and genotypes would not fall into these neat patterns, although you can argue with me that this isn't very neat, but you could see that you'd need to do hundreds and hundreds of plants to try to see if you could get a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio. So that is the independent traits. If they were genetically linked, your genotype and phenotype would fall outside of that expected ratio by some amount. What you'd do is a chi-square test of fitness. But this isn't a statistics class, so we'll move on. Okay, so now we're doing sex-linked traits, and because most of the traits in your sex chromosomes are on the X chromosome, you can think of them as X-linked traits. A classic example is red-green color blindness. So just to jump back before we talk about the color blindness, we just remind ourselves that males have 1 X chromosome and 1 Y chromosome. Females have 2 X chromosomes. And the traits that we're going to deal with here are X to the capital A, which are people that can see red and green. X to the lowercase a, those are people that can't see red and green, they're red-green colorblind. And then the Y is it just means that the Y doesn't have this particular gene, so it can't contribute to the phenotype at all. So the uppercase A and the lowercase a represent the sex-linked trait. And in this case, sometimes you're talking about a defective gene when you say the lowercase a. And so anyway, here's a cross between a parent that the female was a homozygous for having X's because females have 2 X's, but also homozygous for the trait of capital A. let me get rid of, when I did this in class, I actually did a couple different examples on this same slide. Let me get rid of that carrier bit. So the female in this cross is a woman that can see color, and she has 2 good color seeing genes. The male in this cross has his X chromosome has the lowercase a, so this male, and the Y contributes nothing, so this male here is colorblind. So if you take a colorblind male and cross it with a female that has 2 capital A's, the genotype is capital A, you're going to end up with 4 possibilities for your offspring. 2 of the 4 are X and Y, so 50% are expected to be male, and 2 of the 4 are X and X, so they're expected to be female. You mix and match these genotypes, X capital A, X lowercase a, you get a female here that is heterozygous. This female is also heterozygous. This male carries a good gene, the capital A, so it can see red and green. This male received a good gene from the mother on the X chromosome, and so that male can see color. And so that is all 100% of the kids from someone that is colorblind should be able to see color. The exception would be as if the mother was a carrier. But you can mix and match parents and mothers and fathers and see what happens. For example, if we had one of these females here, X to the capital A, X to the lowercase a, we would change that to a lowercase a. And let's say that you have a female that was a carrier and a male that could see color, so he would have a capital A. They would have 50% of their, 50% of their males would have the lowercase a. So if this capital A was a lowercase a, this 1 in 4 children or 1 in 2 male children would be colorblind. Okay, and then the final thing to bring up in this video is polygenic traits. So let's take a trait like height, and when Gregor Mendel studied pea plants, somehow it fit in nicely with a tall or a short. He had some sort of a cutoff for their pea plants, and he just put them into one of 2 categories, and things seem to work out for him. But if we go over to humans, things get a bit messier. And if you look at the height of a bunch of humans, you're going to get basically a bell-shaped curve distribution for height, where, let's say this is females, the majority of women are around 5'6". You're going to have few women that are over 6 foot tall, and you're going to have few women that are less than 5 foot tall. Most are going to be within the 5 to 6 foot range, with the majority sitting close to the 5'6". So the reasons that you get bell-shaped curves like this, there's 2 reasons. One, obviously, is the trait of humans for height is polygenic. At last count, there could be as many as 100 or more genes that contribute to human height, and so it depends on what combination of those genes you get on all 100 loci that determines your height. The other idea is that environment can really affect height. So if a child grows up in a malnourished state, they will not be as tall as an adult as if they had a very nutritious diet with all vitamins, minerals, etc. Those contribute to growing to your full potential. So this is known in genetics classically as the nature versus nurture argument. And so we've got a genetic component that contributes to height, and we've got an environmental component that contributes to height. And so there's an endless arguments about which contributes more, genes or the environment. This is just one of many examples that is argued over amongst scientists and lay people alike. So anyway, that is the extension or the extent of non-Mendelian genetics that we're going to get into for this 100 level class. If you go on to upper level genetics, you're going to learn about additional things like epigenetics and imprinting and all sorts of things like that. Anyway, until next time, this is Life in Biology. Talk to you later.